So what is drug excretion? In a simple definition, it is nothing but the elimination. It's a part, the type of elimination process, which is irreversible removal of drug from the body by all the route of elimination. So elimination is a combination of biotransformation and the excretion process, that is metabolism and excretion process. So all this we went through in the last class. Overall, the elimination is nothing but the combination. It can be the combined process of biotransformation and the excretion process. So when we talk exclusively about excretion of the drug, the excretion word is suitable where the drug is completely removed out of the body. But in case of elimination happening by the biotransformation process, what happens? The drug is getting metabolized and the metabolite will be excreted out of the body. So there is some chemical modification which happens and that modified one will get excreted. So ultimately excretion term can be used when you want to, that is when the drug is completely eliminated out of the body. So now this excretion can be categorized into renal excretion and non-renal excretion process. So renal is kidney, the drug is excreted through kidney, and the non-renal is other than the kidney. So we just revised last class a little bit about individual non-renal excretion process like lung excretion through the lung, where the non-volatile substances are preferably, or the category of the drug which you can expect that getting excreted is the non-volatile. It should be non-volatile in nature. See again, all that non-volatile, sorry, I think I'm using the wrong word. It's a volatile word. It should be volatile in nature, and the drug should be unionized form or more lipophilic in nature. So drug, whichever drug has this property, don't imagine that every drug which are fitting into this category will be eliminated or excreted out of the lungs. But the drug to be excreted through the lungs, the required features are this. So the exceptionals are always there. So these are the examples of the drug which you can expect to get excreted through the lungs, which are anesthetic gases. So volatile nature makes it like easy to excrete out of the lungs. So also we went through the next type category, that is excretion process, salivary excretion. So every non-renal excretion process, depending on which is that pathway, depending on which is that route by which it is getting excreted, the properties of the drug required to be excreted by that particular route will vary. So again, when you talk about saliva, the pH condition matters. So when the pH comes into picture, the behavior of the drug, that is especially electrolyte nature, the drugs which are having the capacity to get ionized and remain unionized. So all that factors will start affecting. So similarly, we understood about the biliary excretion also. This sentence, whatever I actually asked you to rectify, that is a drug with molecular weight of 30 to 300 to 500, is the correct. Actually, I referred it. So I kindly, if at all you have striked it out, you rewrite again. Is if the molecular weight is in the range of 300 to 500. See, bile excretion happens for the drug molecules which are having the molecular weight greater than 500. That is required property for the drug molecule to be excreted by biliary excretion. But if at all it is in the range of 300 to 500, that is, it is less than 500 and near to 500, then these molecules have chance of escaping excreted by both the routes, urine as well as the bile. So these are the examples which I just quoted here, which falls into this category. The range of molecular weight is between 300 to 500. So they have both the options. So also through milk we understood, and let's start with the next very specifically, that is the renal excretion process. Remember always the renal is very specific to the kidney, the organ kidney, where the excretion happens to the kidney. So excretion happening to the kidney is nothing but called as the renal excretion, and it is one of the major excretory organs which we have in our body. Okay, and most of the drug try to get eliminated, that is excreted out of the body by the renal excretion process. But now, as we always try to understand what are the desirable properties. Fine. So one property already we have idea, that is molecular weight. So previously in the bile excretion, we understood that its molecular weight should be more than 500 daltons. And it should be strong polar in nature. That is a required property for a drug molecule. Same here in the excretion by the renal excretion process. the required desirable properties are they should be more polar, highly ionized, water soluble drug again, and excreted related unchanged by kidney. So molecular weight should be mentioned in the next slide. The molecular weight should be, so for a renal excretion of a drug, agents that is suitable candidates are which are water soluble, non-volatile, which is smaller in molecular size, that is less than 500 daltons, and even the metabolized can also, or those drugs which are getting metabolized slowly. So even those drug can also get excreted by the drug because of its molecular. See, there is a chance that drug molecule can have an ability to undergo metabolism. It might get metabolized, even though it is smaller in molecular weight. So don't just again consider that all the molecules which are lesser in the particle size less than 500 will default come into renal excretion process. Not necessary. There are molecules which can undergo metabolism also even though they are below 500. But remember, bile excretion is different. See, metabolism is, bile excretion is different from the metabolism. After metabolism, the metabolite, depending on its molecular size, it comes to the bile excretion process. Okay, where its molecular weight is increased. So the required property for a drug to get eliminated or excreted through urine, that is through the kidney, are they should be highly polar, ionized. Okay, nature ionized, default it makes it more polar, water soluble, and it is excreted relatively unchanged. The unchanged in the kidney, the drug getting excreted usually unchanged forms. If it is getting changed means it is getting metabolized. So its metabolite is getting excreted. So for a drug to be excreted by the renal excretion, so these are the required properties. Okay, and examples, the best examples which you can remember is penicillin, aminoglycosides, and most of the antibiotics. So if they fit into this category, they can easily get excreted out of the kidney. So unchanged removal of the drug from the body through the kidney is nothing but called as the renal excretion process. If it is getting changed, then it is its metabolites which are getting excreted by the kidney. Fine. So at this moment, let me tell you one thing. The urine sample, see there are two sampling techniques we use it for understanding the behavior of the drug in the body. The invasive technique is blood, non-invasive technique is the urine. So at that point I told you, I don't know whether you remember or not, but for a drug, for a urine sample to be considered for my experimental purpose, then we should be sure that the more than 75 to 80% of the drug should be excreted out of the kidney in unchanged form. Then only I can collect the urine sample as one of the sample for my kinetic studies to understand what is happening to the drug in the body. If it is excreted by kidney less than 50%, that means the another 50%, what is happening to that drug? It might getting metabolized. So in that case, I cannot use the urine sample. So for the experimental purpose, if you want to use the urine sample as one of the sample, then you should make sure that the drug has the excretion method through kidney that is more than 75 to 80%. Then only we can go with the urine sample as an alternative sample technique than the that is invasive technique, that is blood sample technique. Otherwise, usually the blood sample is the best sampling procedure which we can adopt. Doesn't matter for us. Okay. Of course, there also we should have the knowledge whether the drug is getting metabolized or not metabolized, but it's a different situation. So right now, let us not worry about that. So for matching to today's topic, remember this. If you want to use urine as a sample for your experimental purpose, the drug should get excreted to kidney more than 75 to 80%. Then only I can go with the urine sample. Otherwise, the alternative is the plasma sample. So now when we talk about the excretion process, that's the renal excretion process, we cannot forget these terms. So which you already learned in your other subjects. So glomerular filtration are different process which happens when the excretion of a drug happens through the kidney. So the three different processes are glomerular filtration, active tubular secretion, and active tubular reabsorption. So three steps you can call it, or you can call it as a three process which happens in sequence. Glomerular filtration is the first, followed by the active tubular secretion, followed by the either active or a passive tubular reabsorption process. So when we talk about kidney as an excretory organ, we should also remember that it does not just excretes the drug out of the drug, it also has other functions. So removal of the metabolic waste products. So during that filtration process or during the excretion process, how that process helps us, how that process helps our body. It helps our body to get rid of the metabolic waste products, which may cause the unwanted toxic effects. See, drug when enters into the liver, it gets metabolized. There's a lot of changes happens to the drug molecule, and that metabolite may be active or inactive state. If it is active with non-toxic effect, fine. But if it is toxic effect, then what happens? It has to be eliminated fast out of the body, excreted out of the body. So that means the metabolic waste product is generally removed out of the kidney. That's the last part when the metabolites try to come out of the body. It maintains a normal fluid volume, which is very important. It balances the fluid volume present in our body. It doesn't allow the um, particles whichever coming or the molecules whichever is coming out of the blood, it is the fluid exchange will happen because of that osmotic pressure, hydrostatic pressure, all that will be well balanced because of the electrolyte concentration or the components which are present in the blood. Everything, all this play important role in balancing the volume of the body fluid. And also it maintains the composition in the body, as I told, electrolytes. Okay, excretes excess electrolytes, water, and the waste product. Of course, well balancing of these electrolytes are very much required because that is the one which is actually exerting the osmotic, results in the variation in the osmotic pressure. Conserves necessary solutes and salts for normal body functions. Fine, it's the same thing, but it's in association with the electrolytes only. Secretes renin, which is very important, which regulates the blood pressure, and also secretes erythropoietin, which stimulates the RBC production. So, other than the function as an excretory organ, the kidney has other functions, important functions, which is very important for our body. So, I'm not going to discuss in detail about the anatomy of the kidney. We know that it's a bean-shaped organ and it is rich, highly perfused organ, and the blood entry and exit happens because of the renal vein and arterial vein, and contains millions of nephrons, all this. Okay, so now nephron, we need to little understand about the nephron structure because that's where actually the excretion process happens. So nephrons are almost like 1.5 million nephrons are present in each kidney. We have two kidney, among the one kidney you can imagine 1.5 million nephrons. So whatever this fiber-like structure you see here, it is nothing but they are the nephrons. So these nephrons are the functional unit in the kidney. The functional unit is the nephron, which is responsible for the removal and the maintenance of whatever the functions we discussed previously. All that functions, whatever we discussed, are with respect to the nephron, which is present in the kidney. Fine. So now we know there is a nephron, something called as nephron in the kidney, and the nephron is a different structural parts, which I just told previously. It's a glomerulus. It has tubules. It has Henle loop. Okay, all this. So anyway, we are not discussing in detail about the structure, but we should understand. As you can see, that yellow color part. This is the main region through which your filtered fluid pass through, and it is accumulated in the form of urine. What's we call it as urine. Okay, how the way that the filtration happens, whatever the drug is carried to the blood circulation to the glomerulus. Okay, that's where the glomerular filtration happens. And from where this blood circulation is happening, it is a network of capillary which is surrounding the nephron structure. As we know that afferent arteriole and the efferent arterioles, afferent and the efferent arterioles. So the drug comes through this, and this drug, when it comes, the blood flows through this region, filtration happens, and that's where the exchange of your drug molecule, electrolyte, everything happens at this place because of the filtration process. And then whatever the fluid comes out here, it is secreted in the form of urine, and which is getting thrown out of the body in the form of urine. So the important features when we talk about the kidney, functions we understood. Now we should also know its other features. Like blood supply to kidney, we've been discussing this from the beginning. Rich but highly perfused organs in our body are kidney, brain, liver, heart, like this. So same here, the blood flow to kidney is very high. So represents 0.5% of the total. The kidney weight is 0.5% of the total body weight, and on an average 20 to 25% of the cardiac output. Whatever the blood comes out of the heart, we try to rush towards the kidney, that is 20 to 25%. Same thing when you're talking about intestine also, there we highlighted one point, highly perfused. Same. So you can, that means these are the regions of our body where you can expect the blood flow is high. So why, how does it matters? It helps in the distribution process. The blood supply is so much important because it is the one process which makes the drug to distribute throughout the body. And same, the blood flow also take part in the excretion process also. So it is supplied by the renal artery. Okay, all these are little basic informations. The blood is supplied by the renal artery, which is further subdivided into interlobular arteries, which penetrates within the kidney and branch further into afferent arterioles. So let me go to this one. So this afferent arteriole, whatever we have labeled here, and this is the branches, sub-branches of this renal artery, which carries the blood towards the kidney. And this afferent arteriole carries blood towards a single nephron. So each and individual nephrons are actually surrounded with a network of capillaries. So afferent, right now I remember afferent arterioles carries the blood towards the nephron, individual nephron. And same, this afferent arteriole, whatever, once it comes to the glomerulus, it further sub branches itself, and then it gets uh, it covers other parts of the nephron. So that's called as the efferent arteriole. The further branching, that's what, from the capillaries with the Bowman's capsule, the blood flow out via the efferent arterioles. So blood comes through the afferent arteriole, goes out through the efferent arterioles, and then into the secondary capillary network. So secondary capillary network is the one which is actually surrounding the tubules. So why it is so important for us to know this important network of the capillary is because if something happens in the nephron. One is filtration, okay. The second one is the active secretion. In both the cases, from the blood, the molecule try to enter into the nephron tubules. In the active secretion also, the drug is getting, the drug molecules try to move out, or any molecule try to move out and goes into the that is nephron tubule. But in case of, when you come to the reabsorption process, something happens other than the secretion and the filtration. The last event which happens is the reabsorption, tubular reabsorption. So there is a chance that the drug would have filtered here, okay, and because of some other reason, there is a chance that it get reabsorbed back to the blood. So that's what makes the fluctuation in the plasma concentration. There may be many reasons for that, the urine pH, or maybe there is some active molecules, carriers which are present there. So that is a reabsorption process happens. You'll understand when I just go ahead with a few more points. So right now you remember, it is highly perfused organ, 25 to 20 to 25% of the cardiac output is rushing towards the kidney, and the blood is carried with via the renal artery, which is further subdivided into afferent arterioles, and after that it is further branched into the efferent arterioles. Afferent to efferent, A and E. That's the only difference in the letter in that afferent and efferent arterioles. So these afferent arterioles are the one which actually surrounds the tubules, and that's when the reabsorption process happen there. And because of which the drug molecules or any other molecule can, or electrolytes can get reabsorbed into the bloodstream. So that's little bit about the anatomy of the and the functions of the kidney. So coming to the renal blood flow, certain terms we have to remember, like as we see in this slide, renal blood volume. Okay, RBF it is called as. It is the volume of the blood flowing through the renal vasculature per unit time. That is 1.2 liters per minute. So 1,700 liters per day. So how much volume of blood is flowing to the kidney? So how does it matter? It matters because the elimination of the drug is mainly affected by the blood flow. So normal person, if nothing has happened to the kidney or there is no heart disorders, then everything is normal, fine, then the blood flow will also be normal. In the normal condition, this is what the value of the RBF is. Renal blood flow volume will be 1.2 liter per minute. So same way, there's one more term we can understand, that is renal plasma flow. Okay. So when we say blood, it is a plasma in combination with all other components of the blood. That is your RBC cells or your other proteins which are present. Fine. So when you tell plasma, it's the renal blood flow minus the volume of red blood cells. So we are removing that value and we are converting that into the renal plasma flow. So then that this small calculation what you can see, okay, where they are removing the hemoglobin content and finally they got the value 0.66 liter per minute or 660 liter per day. So when you take 660 liter per day, it is the renal plasma flow. When you take 1700 liter per day, it is a renal blood flow. So plasma flow and the blood flow. These are the two terms which can be used to understand the flow of the blood towards the kidney. So coming in, next one is the mechanism of the renal excretion. So I told you three major process happens, which happens in the nephron, especially in the nephron part. Filtration, which is called as glomerular filtration. And next one is the active tubular secretion and the tubular reabsorption. Also, our active tubular reabsorption, which is one of the event which may be happening depending on the carrier. But generally, it is just called as tubular reabsorption, which can be passive or active process. So 5 marks question can be expected with respect to mechanism of renal excretion. So let's discuss with the first one, glomerular filtration. Look at the term, it's the filtration. Remember, filtration is a process wherein which what happens is depending on the size of the particle and size of the pore that is through which the particle or the fluid need to pass through, the solid particles get separated from the liquid. So it's nothing but it's a mechanical process. Depending on the pore size and depending on the particle size, the filtration will happen. And most important thing is it is unidirectional. See, reabsorption, I told you, tubular reabsorption is the opposite direction to the glomerular filtration. In the glomerular filtration, the drug molecules, whichever is smaller than the pore size of the glomerular, that is glomerular, that will pass through. If it is bigger in size, it is retained. That is unidirectional. You can't expect the filtration happening in the opposite direction. So it's a first point to be remembered is the glomerular filtration is the first process which happens in the nephron, okay, and which is a non-selective unidirectional. What is this non-selective? See, when we say active process, anyway, active absorption or active secretion in case of kidney, they have carriers. So when we say carrier, they are selective. But glomerular filtration is a mechanical process, so where it depends just on the pore size of the membrane. There is a glomerular as well as the particle size. So it is non-selective. Doesn't matter what type of molecule it is, just the size matters. So smaller in size and spherical in shape are the one which can easily pass through, but not high molecular weight, which are larger in size or long chain molecules. So anything very larger in size, okay, cannot pass through, or bigger than the size of the glomerular membrane, it cannot pass through. So most compounds, ionized and unionized, are filtered except. So it doesn't matter whether the compound is ionized. That's what it is, non-selective. It is unidirectional. It doesn't matter any other properties of the drug molecule. It's only the size. Okay. So compounds, whether it's ionized or unionized, both can be filtered, except those are bound to plasma protein. So you can now correlate whatever we have understood in the distribution chapter or the plasma protein subtopic, drug protein binding topic. There also we learned that drug binding to the plasma protein increases its complex size, and because of which it cannot cross the capillary membrane. Same here, since it cannot cross the capillary membrane, filtration will not happen, that is in glomerular. Okay, so most compound, if it is unbounded, in the unbounded status, then it can easily get filtered if the molecule size is less. So filters, so the irrespective of this, that's what I was telling you, irrespective of the charges, irrespective of the nature of the molecule, if it is less than 500 Dalton, the glomerular filtration definitely happens to that molecule, drug molecule. So now, by chance, if the molecular weight is high, but now one more factor can influence the filtration, that is the structure or the shape. For example, if though the molecule is higher in its molecular weight, if the shape is linear, just like a thread, okay, then it can easily squeeze through because by the filtration process. Though the molecular weight is high, but the structure is, is the one which is influencing. Then even for the molecule above 400, so shape of the molecule becomes the determinant factor. So the important factor which governs at that time is the shape. So linear molecule with a high molecular weight can also easily pass through the glomerular filtration because it can easily squeeze through the channels which are present in the capillary membrane, also present in the glomerula. Okay, so glomerular filtration drug is directly related to the free, means the factor. It is directly proportional, or yeah, I can say proportional also. As the free drug concentration increases in the plasma, GF, that is the glomerular filtration rate of a drug, will also increase proportionately. Okay, free drug is unbound drug. So this value, when I exclusively calibrate the value, the filtration rate, for a normal person, it is 125 ml per minute. Please behind this number is important because this is the value, this is the number which gives us an idea whether the kidney is functioning normal or not. So there are different methods by which you can actually determine the rate. So in that, when you determine that rate and if you find that value is fluctuating, either negative way or in the positive way, that gives some indication that alarm that there is something wrong with the kidney function. So that's the reason I'm telling you I'm giving you idea about these numbers. Even in case of the blood flow rate also, even that is also important information which helps us to understand the condition of the person. So for a normal person, the blood flow, the glomerular filtration rate is 125 ml per minute. So and look at this number, 180 liter, the next point, of the fluid is filtered. So you can imagine our kidney, how much it is working. 180 liters of the fluid is filtered. But see that the last point. Even then, the urine, whatever we send out of the body, which comes out of our bodies, approximately 1 to 1.5 liter. So it is too less when we talk about this 180 liter. And this is mainly because 99% of the fluid is reabsorbed. So reabsorption is very important process which happens in the nephron. I now hope you understood that reabsorption is another process which actually keeping our body fluid, required normal body fluid volume. If the reabsorption is not happening, what happens in the glomerular, in this structure, the filtration might happen because this is only based on the pore size and based on the particle size. Fluid can easily comes out of the glomerular. But if that is not reabsorbed back, you can imagine how much water we should have been drinking continuously in order to maintain our body fluid. But that is not happening because whatever fluid comes out here is getting reabsorbed back to the body. So that actually balances the body fluid in our body. So that's what I'm trying to say here. Though 180 liter of the fluid is filtered, so that approximately it is 1 to 1.5 liter of urine is filtered out, and that is mainly because maximum volume of fluid is reabsorbed back into the blood circulation. Okay. So now just to wind up, factors influencing the glomerular filtration. What are the factors? Already we got an idea now. It's mainly the particle size. So total surface area available for filtration I cannot neglect because the surface area of the glomerular that unit at the nephron part, that is the first part of the nephron. So that surface area is high, even that area matters. Okay, and permeability of the filtration membrane. It's all the very general factors actually. How permeable? Because anything goes wrong pathologically, something happens to the kidney, there is a chance that the surface area might get affected. Also, there is a chance that because of that change in the surface area, the filtration rate will reduce and also the permeability might affect it. So, in the pathological condition, there is a chance that the area of the glomerular as well as the permeability capacity of the unit both can be affected. That's the reason we consider these factors. We cannot neglect. So, net filtration pressure, that net filtration pressure is mainly because of the movement of the blood which occurs towards the kidney. That is a hydrostatic pressure. So this capillaries, we already discussed this. Most of our body part, the capillary membrane are highly permeable in nature because of its greater surface area. So the driving force for the filtration here is that is a glomerular unit is the hydrostatic pressure of the blood flowing in the capillaries. So everywhere we learned about the driving force. So here also the driving force for the filtration force is the pressure exerted by the blood or the pressure difference. The pressure difference between either side. That is one side is the capillary pressure, that is the hydrostatic pressure. The other side is the fluid which is present in the glomerular unit. Okay, because of the change in the pressure difference and also the hydrostatic, there is a blood flow rate, everything results in the filtration process. So the driving force for filtration through the glomerular is the hydrostatic pressure through the capillaries. So overall, the factors influencing, maybe at this point I missed out some points like particle size. You can add up that factors influencing glomerular filtration is along with this total surface area. This points are very specific to the anatomy of the that is nephron. So you can add up to this even the particle size and the shape also are also the factors which can affect the filtration process. If they ask you to discuss about the factors affecting the glomerular filtration process, add up those 2 points also. Now, as one additional information, I told you why the rate, we should remember that number 125 liters per minute, sorry, ml per minute, that value. So, as I told, in order to understand the functioning of the kidney, there should be some procedure for that. So, in order to understand the glomerular filtration rate, so they try to determine this filtration rate and they do it by using some like what you call that indicators. I don't know, I'm using the right term, but we use some substances which are exclusively get excreted by the kidney through the filtration process only. So these are the three substances which you can imagine. You can consider mannitol, sodium thiosulfate, and inulin. These are three components which can be used as an indicator. Of course, I think we can use that word indicator because these are the one which are indicating the functioning of the kidney. So this, they are administering the known amount of this mannitol or whichever you are seeing as an example. They try to collect the urine sample and they try to measure the amount of mannitol coming out or the sodium thiosulfate by analytical procedure and they will confirm the volume. So that's how you determine or you try to understand the function of the kidney or the working condition of the kidney. Creatinine also can also another reason, but somewhere I got the information that it is used mainly to measure the GFR in the dogs. So anyway, but mannitol is well known now. They use mannitol to understand the filtration rate of the nephron. So coming to the next one, is the next process. So understand it's the word active. So wherever the word active comes into picture, there is a carrier. So it's a carrier mediated system. It requires energy and against the concentration gradient. This will make you to remember all those mechanism we learned in the absorption chapter. Same thing here. The mechanism is same. It's structural dependent. Two molecules will compete for the same carrier. Everything applies same. Whatever the information, the knowledge we have gained, the same knowledge is applicable here. So only thing is that it's happening in the kidney, specifically in the kidney because of the carriers which are present in the kidney. So it is transported across the concentration gradient from blood capillary across the tubular membrane into the renal tubule. Okay. So this one is capacity limited process and may be saturated. So all this point makes it, I mean, because the drug molecule is dependent on the carrier, the entire process is called as active tubular secretion process. Secretion word indicates that the molecule is moving from the blood into the nephron tubules. Okay, it's adding up to the urine. Fine. So there are two systems, active and the renal secretion systems can be categorized based on the nature of the drug. Systems for the weak acid and system for the weak bases. It's depending on the carrier and depending on the nature of the drug molecule. They have understood that there are two types of secretion systems are present in the active secretion process and can be categorized into weak acid